Hi, everyone. Welcome to our Thursday webinar series. My name is Lourdes. Glad to have you with us today. As you may have seen in the waiting room slide, today's webinar is sponsored by the Bloom family in memory of Alan Bloom. Alan's wife, Janie, wanted to share this message about our family's choice to support educational opportunities for the CCF community. Today, we are sponsoring this webinar on CAR T-cell tumor therapy because we believe in supporting patients and families with the best opportunities to enhance and extend the lives of patients with cancer. We learned about CAR T-cell therapy from a family friend who is a pathologist. He believed that because Alan's cancer had spread to his bone, that CAR T-cell therapy for tumors could have potentially extended his life or saved him. However, that was not available at that time. We hope that this therapy and others continue to be researched and expanded and save as many lives as possible. This webinar honors the wonderful man he was, the husband and father, grandfather, and friend we love and miss so much. Thank you to Jeannie and the Bloom family for their continued support of the Cholangiocarcinoma Foundation in Alan's memory. I want to remind all of our viewers to please put any questions that you might have in the chat or Q&A, and to please make sure that those questions are general in nature, as our speaker can't answer your very specific medical condition question. And with that, I would like to introduce our speaker today. Dr. Gregory B. Lazinski is the John Coffin Family Professor for Pancreatic Cancer Research and Vice Chair for Basic Research in the Department of Hematology and Medical Oncology at Emory University. He serves as the Associate Director for Basic Research and Shared Resources for the Winship Cancer Institute, where he provides oversight and direction for development and growth of basic science activities across the Cancer Center. The Lazinski Laboratory is translating novel immune, cellular, and targeted therapies into clinical trials for patients. Dr. Lazinski was elected as a special expert in immuno-oncology and translational science for the Hepatobiliary Task Force of the National Cancer Institute Gastrointestinal Steering Committee. His team has been pursuing combined therapeutic approaches targeting cytokines and signaling pathways derived from the tumor and stroma to enhance the efficacy of immunotherapy. This work has fueled many collaborations, including funded grants in pancreatic cancer and cholangiocarcinoma. Dr. Lazinski is on the Scientific and Medical Advisory Board of the Cholangiocarcinoma Research Foundation and has had continuous NCI funding since 2008, citing over 140 peer-reviewed publications. Thank you for being here.
My pleasure, and thank you for the invitation and really the opportunity to interact. I've been very pleased to be involved and integrated within the Cholangiocarcinoma Foundation, and thank you to all the donors and others that support the work. Really, everything that you guys do is absolutely wonderful and definitely making an impact. So today we'll talk about immunotherapy, specifically cellular therapy, and how what we do is hopefully weaponizing the white blood cells. I had to come up with a creative title to get your attention, and hopefully I'll do a good job today. Let's see. So in order to really make an impact in rare diseases like cholangiocarcinoma, it takes really a lot of different people working together. And this is one of the great things about the community, is that on both the scientific and the clinical side, as well as patients, caregivers, advocate, and the foundation, everyone does indeed work together, and it's a internationally collegial environment where we all are marching towards the same goal of making an impact in this disease. And this photo is from our 5K event that our cancer center puts on, and the disease does get close to my own home. When I moved to Atlanta to take on this opportunity at Emory about 9 years ago and lived in our neighborhood, I met this woman in yellow. Her name is Elizabeth, and she is a cholangiocarcinoma survivor for more than 10 years. And my son is next to her with one of his friends, that's her son. And, we got into conversations around the pool, and it, usually starts in very general, hey, what do you do for work? And it's usually very, hey, I do research. What kind of research? Cancer research. Oh, what kind of cancer? GI cancer. And then you go deeper down and down the path, and it turns out, I opened up a little bit and said, well, our lab works on cholangiocarcinoma. And she said, oh my goodness, I'm a survivor. And I do have her permission to share this picture. It's obviously very inspiring to meet individuals like Elizabeth and others that remind us why we're doing what we're doing, in the hopes that we can have many more examples of people living long and beating this disease. So we have a great group of collaborators. Most notably, Johns Hopkins has worked very closely with us over almost a 7 or 8 year period, a number of different active projects and things that have gone into large national clinical trials. So quite a bit of action in this space, and I'll tell you about some of that today. So just by way of outline, what am I going to talk about? I think it's good to just get everyone on the same page at first and set a base level of understanding about what is your immune system and what are white blood cells and what is their role in infection, in cancer therapy. Certainly, it's important in cancer immunotherapy, which is the second thing we'll talk about, different types of immunotherapy. And then third, we'll get into the more fun stuff, and that's how we are doing what we call weaponizing the white blood cells and give an overview of different types of cellular therapy, including CAR T cells. And then I'll finish with a few examples of some things that we're doing in our own collaborative research group at Emory and beyond, some emerging ideas related to cell therapy, specifically in cholangiocarcinoma. And then hopefully, if there's time, we can chat about the best way to integrate this approach for patients in the clinic, and then maybe a bit about the future for cell therapy in cholangiocarcinoma. And to really appreciate the disease and where we're coming at it, it helps to take a big picture view, right? So this slide on the left shows what a pathologist would look at under the microscope if they're considering a cholangiocarcinoma tumor. We know traditionally that cancers are cells that have lost their normal regulation of growth and proliferation, right? But a cancer or a cholangiocarcinoma is not just a ball of proliferating cancer cells that have gone out of control. There's actually quite a complex environment in these tumors. There's things like blood vessels. There's other cells that are called fibroblasts that provide structural support to the tissue in this tumor lesion. There's also cells that are white blood cells or immune cells intercalated within these tumors, not just in the primary cancer, but also when it spreads. And many of us, including myself, have data and really strongly believe that this really intimate interaction between the cancers and the white blood cells that live within them is instrumental in their growth and progression. And our goal is to try to do what's on the right-hand side of the slide from a BBC article, and get those immune cells shown in orange to fight and engage in battle with these cancer cells and win. And this is a picture of a T cell that is what we call cytotoxic. It's literally spitting granules inside of this cancer cell to hopefully pop it like a balloon and release its contents into the environment and really win this battle. And again, that's our end game, that's our goal, and what we're all trying to do. So to back up a little bit more for those who haven't been educated in biology or hematology in this case, white blood cells can take on a number of shapes and sizes and functions, of course. We know they all arise from a common stem cell-like progenitor that typically lives in the bone marrow of the body. And then once it gets the right signals, it can go down a number of paths and differentiate into different types of cells that all meet functional needs of a human being or an animal. And these cells, the ones we classically think of as white blood cells, are shown in the red block box at the bottom. They all look very different, and that's because they have different individual functions. So plasma cells or B cells, they make antibodies. That's what protects us from things like the cold or the flu. Other cells like T cells or NK cells, which stands for natural killer cells, many times they have specialized functions that when a cell is infected with a virus, it's seen as foreign and they go in and they literally kill it. We want these cells to act effectively against cancers and do the same thing. They have supporting immune cells or white blood cells that are shown here that adopt drastically different shapes and functions. Things like mast cells are involved in allergy, while things like dendritic cells and macrophages have their own sorts of functions. So they are very distinct, but again, all arise from a common progenitor, and collectively we know and love these as white blood cells of our immune system. And, depending on what their structure is, we like to think that their form meets their function. So take the natural killer cells and T cells, for example. This is a computer rendering. They have little dots in here. These are actually these cytotoxic granules that you can see in a real image of a cell here. When it engages a target, like a cancer cell, and it functions effectively, it will directly contact the cancer cell and release those granules inside it, again, popping it like a balloon. Other cells, dendritic cells, they have these projections or dendrites, hence the name, that facilitate communication with other cells and driving productive immune responses. We talked about the plasma cells, for example, and you can see this is their nucleus in the dark part, and then the surrounding areas of cytoplasm. This sort of empty room here is where all the proteins are made. And since these cells' job is to make a bunch of antibodies, they need room to do that. That's where they're made, they're spit out, and they circulate into the body and protect you from infection. Finally, we have the macrophage, and these cells are known to engulf or eat things. Phage is sort of synonymous with eating, and this particular cell is of the lineage called a myeloid cell, and these types are really, in some cases, seen as bad guys within the cancer. And they're one of the cells that, in the right conditions, can drive the cancer to progress and are something that we're quite interested, as well as others, in targeting. So again, they each have their own jobs, and this really orchestrates the different types of immune responses that someone can have. So other terminology that's important as we appreciate how cancer can recognize or be recognized by the immune system is this concept called an antigen. So an antigen is just a fancy name for any protein that will bind an antibody, for example, that comes from B cells or other things from T cells. And these are essentially proteins that are present on things like viruses or bacteria that allow your body to see it as foreign and recognize it immunologically. Antigens or proteins that are abnormal are also found on cancers. But when your normal cells have their own antigens or proteins recognized by the immune system, it can lead to harmful immune responses like autoimmunity. And those are the types of things that we avoid. Now, this concept of recognizing a specific antigen is the foundation of what we know about the thing called memory, right? You get a vaccine, it usually takes a couple weeks to kick in and protect you, like the flu shot. That's because your body needs time to make these proteins that protect it. But at the end of the day, you'll have immunity and long-lived what we call memory protection, and that's shown in the red curve. In contrast, there's other immune responses that are very short-lived and quick. They don't have any memory, but like if you get stabbed in the arm, it inflames and you have a really robust, fast wound healing response that tries to heal it. These are things called innate immune responses in your body's natural reaction to injury that isn't associated with a memory. And these sorts of things are both important and they interact with one another, but kind of the foundations of general immunology. So these concepts with using white blood cells have absolutely been adapted into treating cancer. And this results in the larger field known as cancer immunotherapy. There are a number of different types of immunotherapy. One of the first that ever was used is this idea of what we call an exogenous cytokines. And that is really a fancy way to say immune hormones made by your body can be engineered in a lab and re-infused into a patient to essentially trick the body into thinking you're having a fever, and it turns on all the white blood cells, gets them really revved up and activated, and it goes in to kill the cancer if it works well. And this approach was used quite extensively in the setting of melanomas and other types of tumors and was quite effective in certain populations of patients. There's other types of immunotherapy that you've probably heard of called T cell checkpoint modulation or immune checkpoint blockade. These are things like PD-1 or CTLA-4. The trade names are Opdivo or Yervoy. You've probably seen commercials for these when you watch television at night. But really, if you think about a T cell, these are the ones that have cytotoxic capabilities. There are surface proteins that stick out and are visible that when you engage them, they can either turn on the T cell and make it grow and highly active, or others that are on this side that are inhibitory. So if you engage these markers like PD-1, it will slow down the T cell and essentially shut off its function. So these we'll talk a bit more about. Other people have worked quite feverishly on cancer vaccines where you can encode some of these antigens that are specific to the tumor cell, give a shot, and then get a robust immune response that may have some benefit. Other people are using this idea of antigen and the fact that another protein called an antibody can recognize it specifically, and then they can engineer this protein and attach chemotherapy agents to it so that it can selectively go only to the tumor, bind to the tumor cell in a specific manner, and deliver that chemo or other molecules right to the site where you want it and have an impact. Then finally, kind of what we'll focus on today is this idea of using T cells themselves and transferring them back into the body after they have been engineered in a manner that allows them only to recognize a component on the outside surface of a cancer cell. And we'll talk more about CAR T cells or TIL therapy or other modalities, but this is what they look like inside the cell as well as the outside of the cell where they bind their target and then do all sorts of things downstream. So that's the overview of immunotherapy. This is no secret that it has been very impactful. In 2018, Doctors Hanjo and Allison, for their discoveries related to immune checkpoint blockade that I talked about, received a Nobel Prize, and very well deservedly. And one of the things that they learned is really how this immune checkpoint blockade process can wake up white blood cells. And the mechanisms by which this occurs are shown in a schematic here, where in green we have a white blood cell, a T cell with, again, these cytotoxic granules, and it will recognize these proteins or antigens that are presented to them. They're handed off to them by other cells, including tumor cells. And when all things are going well, these T cells will make more of themselves, they'll grow, they'll produce these cytotoxic killer molecules, and they'll mount effective immune responses. But in the setting of a cancer where these T cells live among the tumors, they're constantly encountering the cancer cell and they're kind of having a failed attempt to eliminate it. And long story short, they get tired, they get exhausted, in fact. And one hallmark of a T cell that gets tired and exhausted is the expression on its surface of another marker or protein called PD-1, which is shown here in green. So when this occurs, the T cell in essence gets turned off, and all these things that it does that might mount an effective anti-cancer or antiviral response even get turned down. So what people like Doctors Allison and Hanjo discovered were some mechanisms by which you can block these inhibitory interactions and really wake the T cell back up so that it can be active again and go in and fight the cancer and eliminate it. And it's actually been quite effective across a number of different solid tumors and even made some impact in the setting of cholangiocarcinoma. So in a really nice summary review article by my colleague Tim Gretton, who I've met through the Cholangio Foundation and a fabulous scientist at NIH, he has a good summary about all the different types of immunotherapy that are being used in the setting of cholangiocarcinoma, including in lab studies as well as in early clinical trials. And these consist of a number of different treatments, including antibodies, some of the checkpoint inhibitors we've talked about, vaccines, and then the topic of today's talk is this idea of adoptively transferring T cells back into the body. And there's many different types of this therapy alone that are being investigated, and we're really quite hopeful that we can make some headway in this space. And I'll hopefully give some encouragement that there are some wheels moving in the field in this realm today. And why? We have to do better. The FDA approval of immunotherapy in cholangiocarcinoma has happened, largely due to two pivotal trials, the TOPAZ-1 trial, as well as the Keynote 966 trial. These are both published, and what they show is that you can give antibodies that wake back up the T cells. They hit these PD-1 or PD-L1 targets, and they are given with chemotherapy, and patients receiving them do better than receiving chemotherapy alone. So these are overall survival from this clinical trial, a large trial that was published a few years ago. Patients getting immunotherapy on top of chemo are in red, and you can see they have longer survival as compared to those just getting chemotherapy alone. So this is important. It is impactful, but you can see that we have a lot of work to do still. There's quite a big area above the curve, and patients need therapies, and particularly those patients who get this in the front line, which is often given now, they need other options. And all the patients now that have received these will likely, go on to other clinical trials and be resistant to some of the common immunotherapies. So we really have to put on our thinking caps and get data and work with a bit of urgency to identify new ways to intervene in this process. So that brings me to the question is, what if we could get these white blood cells from the immune system to selectively recognize only the cholangiocarcinoma tumors and then give them back to patients? And this very idea is the foundation of what we're calling cellular therapy. And we'll spend the rest of this hour talking about that. So we all know our friend who's on the call, Melinda Bikini, who's an inspiration, and she's, one of the things that drives so many of us. Her own story with cellular therapy. This is a manuscript that describes her journey, initially published in Science. Eric Tran, who was at the time at the NCI, now has his own fabulous productive lab in the great Northwest of the United States, described how these tumor infiltrating lymphocytes or white blood cells can be selectively target against a marker on her tumor and given back and have dramatic clinical responses. And this is sort of the thing that we want to strive for, is more survivors given aggressive cellular therapies and done so safely. So, this is one of the most famous examples, and the field continues to evolve. So if you look at what this process is, this is, again, adoptive cell therapy. And in Melinda's case and others, what we do is we use tumor infiltrating lymphocytes, or TILs. And these cells are now FDA approved in the setting of melanoma. The process is illustrated here, where a patient would have a tumor. It's taken out by a surgeon and then expanded in the laboratory, where only the purple cells are those that recognize the cancer specifically and not all the other normal components of the body. So we have ways in the laboratory we can selectively expand these and see you have all the purple cells left, and then they're reinfused into the patient to hopefully have a very dramatic anti-tumor response and elicit long-term survival. In the setting of melanoma, this led to an FDA approval just a little over a year ago, where again, you're looking at a survival curve where the patients receiving a TIL therapy compared to a standard immunotherapy had a very prolonged and significant improvement in progression-free survival. And if you look at the lesions that they were measuring by radiology for the patients, nearly half of them had a shrinkage of their tumor. So very dramatic responses in those that received the cell therapy compared to immunotherapy alone, that again led to the FDA approval. And there's a number of active studies going on to improve the specificity of these cells by finding ways to enrich them to only recognize the cancer. So again, very impactful and starting to, get into other solid tumors in a more widespread way. One of the reasons for success about with these types of cellular therapies, including CAR T cells or TILs, as I showed in the prior slide, is the fact that there's another step I didn't talk about. And that's before the patients receive the new cells, you have to essentially make room in the body to receive them so that they can go into a healthy environment. They have room to expand and grow and hopefully become a greater proportion of the total white blood cells that circulate will be skewed towards recognizing components of that cancer that you want to get rid of. This process is called lymphodepletion. It can be done using a combination of different chemotherapy drugs. It's done routinely in patients with blood cancers as well as those getting transplants, for example. In animal models, we can use low doses of radiation to do the same thing. And it does a number of things. I won't go into the weeds here, but in addition to making room, it may also get rid of some of the bad cells that support the cancer, which have accumulated over time in larger numbers. And this sort of wipes them out and sets the stage at a more even playing field for those cells that you're intentionally giving to come in and take hold. So that's the process of lymphodepletion, and it's important and I'll get into some of our early data later about this. So what are CAR T cells? These are different but similar to the TILs that I talked about before. And one of the advantages of a CAR T cell, which stands for chimeric antigen receptor, it's a big series of words, but I'll tell you what that is in a minute if you don't already know. You can actually get the blood from the circulation or the arm of a patient. And instead of undergoing a surgical procedure, as you might imagine, this is a lot safer in many ways and more attractive. You just have the normal circulating T cells. You can then do something really funky and take them into the laboratory, and using a viral approach with a virus that importantly doesn't replicate, so it's relatively safe, you engineer the T cell to express on its surface a protein that recognizes only components of the cancer. Um, so this is called a chimeric antigen receptor, and it's a chimera or a mix of two things because one half of it actually looks like an antibody, the red half, while the blue part goes down inside the cell and sends um, growth signals to that T cell so it makes more of itself. And by this process, after you've made the T cells recognize what you want them to, you can grow them up by the millions in the lab. And then you lymphodeplete the patient again, as I said, and put the cells back in. And when this goes well, you can see that the CAR T cells that have been redirected only against the cancer cell recognize it. So you have these little red things binding the green nubbies hanging out of the cancer cell, and these are the antigens you want to target, and this cancer cell will be lysed because the cells are really going in after them and trying to kill them. So this is the essential process for CAR T cells and how we can engineer them. So what's happening in this field? There's quite a bit of activity. This slide's even a bit outdated. It was from 2022. And this data is not specific only to cholangiocarcinoma. This is across all types of cancers where you can see there's a number of clinical trials in phase 1, early phase, all the way to phase 3 that are looking at testing these cellular therapies for patients. There's other types of cell therapies as well. Things called a T cell receptor that you artificially put in that has their own advantages. People are using these natural killer cells and TILs and many other types. This market is really expanding, and we're hoping some of these things work so that we can give opportunities for long-term tumor control, or even, dare I say the word, cure in some patients. So a very exciting space. In cholangiocarcinoma, if you look on the surface of the cancer cells, this is kind of a menu of what people are currently designing cells to go after. And you see some of the things you may have heard of, like mesothelin, HER2. There's also some more innovative targets, and you can see this chimeric antigen receptor engaging the T cell on the inside and binding the cancer on the outside, with the goal that it will stick there and it will kill it, and then it can move on and do what we call serial killing, and one after the other knock down the cancer cells. And this is really the dream for which we want this therapy to take hold and work. And another view at these chimeric antigen receptors, again, you have a virus you put actually in the T cell. The virus does not replicate, so by and large, it's quite safe. This is all done in the lab, and then the T cells have this cargo that sticks out on the surface. It selectively interacts in a very specific manner with the tumor cell, and when things go well, it can actually lyse it. So a very neat and cutting edge technology that is what we call weaponizing the T cells to, again, selectively recognize and hopefully eliminate those cancers. So we have a lot of potential, but the fact of the matter is cellular therapies don't have magical results in every single situation. And some of the reasons for that are listed here. I'm not a fan of big bulleted lists, but this is one time where I think they're useful. You can think about how, tumors are like viruses. They undergo an evolution. They're very smart. So the very thing you engineer the T cell against can actually be downregulated on the surface of the cancer, or it can be cleaved off and it's no longer visible. So those CAR T cells that are meant to recognize a target now have nothing to bind to, and the tumor has evolved. So that's a problem. We all know that it's actually quite hard to get T cells to go where you want them to go in the body, right? You have to get the cells to be given by the vein or some other means, and then go all the way through the complex network of vasculature in the body and somehow magically get to the tumor, right? That's quite a challenge if you think of the landscape and the real estate they have to navigate to get into the tumor. So that's a challenge. Let's say they successfully get to the tumor then. Cholangiocarcinoma is a hostile environment, right? It's like putting a T cell in the middle of a hot volcano. There's a lot of barriers there to get it to stay around and survive. These tumors lack oxygen. They have a protective, like fibrotic, collagen-rich ring often that surrounds them that it's hard to penetrate and get cells or drugs in. And then finally, another challenge with why this is so hard is that some of these targets we design CAR T cells against, or other types of T cells, they're not perfect, right? Some normal cells can actually express them, and that's a concern for these things called cytokine release system or other types of toxicity, where you have the T cells inside the body going crazy and trying to kill stuff, but they're recognizing the wrong thing. So we absolutely don't want that because that can lead to toxicity, and we're trying to do the opposite. We're not trying to injure people. So our lab and many others are thinking about what the best possible solutions are. And there's a number of things that, excuse me, are undergoing research and we can do. The first is by changing the type of white blood cell that we use as a template to change. Usually you take out all the blood and engineer the bulk T cells that can be many flavors of T cells. We'll show you a little bit of data indicating we may have honed in on one very specific type of white blood cell that is kind of a superhero, and we think we can engineer it very effectively to eliminate cancer. There's always a need for new targets on the tumor, right? We can do better than we're currently doing with things like mesothelin and HER2, and a number of groups are going after that and even finding personalized ways to find each patient's own individual unique target. We've got new strategies to engineer these white blood cells to resist some of these exhausted phenotypes. And then finally, the trend in the field that we're very interested in is this idea that maybe you can give T cells together with a drug that addresses one of these really hostile mechanisms that resides in a cholangiocarcinoma tumor. So we'll talk a bit briefly about that. So there have been some advances made in this idea of combination therapy, specifically in GI tumors, including cholangiocarcinoma. So this again is our process of getting TILs or tumor infiltrating lymphocytes. Again, take out the tumor, grow them up, and put them back. This work was done by none other than Dr. Rosenberg and his team at NCI, just published a few months ago in Nature Medicine, a really prestigious journal, where they did the thing where they combined this TIL therapy that recognizes antigens on the tumor with immune checkpoint blocking antibodies called PD-1. And their results were pretty impressive, all things considered, given where we've been in the field. These are some of the tumor curves from the patients. Actually, 6 of the individuals on this clinical trial had cholangiocarcinoma, 2 of which had a partial response. So there was some activity. And the cells shrank some of the tumors. And 25% of the patients, a quarter of them enrolled who were, had really horribly aggressive cancers, had responses. So this is definitely a step in the right direction. But, again, we have work to do. There was not a 100% response rate. Not everybody had durable responses. And we're trying to hone in on why we think that might be, and many labs are, including, I'm sure, Dr. Rosenberg's. And one hypothesis or thought that we have is that it's this really hostile environment that exists in the cholangiocarcinoma or other GI cancer tumors, where they're preventing the best T cell you can make to do its thing once it reaches the site. And we'll talk a little bit about that. And what's wrong with these tumors? Why are they so nasty? Well, this slide illustrates some data on the left out of our lab that my talented postdoc made. This is a microscope image of a normal liver on the left and a cholangiocarcinoma tumor on the right. And if you know nothing about science, hopefully the way your room is lit and your screen, you can see that there's more green over here. And all these fancy letters and numbers in the middle just correspond to different surface proteins present on immune cells that are of this hostile myeloid lineage. And we have identified different targets, and we know that the cancer has more of these nasty myeloid cells that we think are fighting the T cells that you put in. Other groups, I collaborated with a great group of investigators at the University of Rochester, led by David Linehan. They stained some patient tumors, and they have a marker that the pathologists use to identify cholangiocarcinoma called CK7. You can see it's brown, which means it's positive. And then they have another marker on the same tumor called CD45 that stains for white blood cells. So you can see that in these tumors, there's also white blood cells, again, having this conversation like I showed on the very first slide. And if you further drill down deeper and ask what the identity of these white blood cells are, they're the same ones that our lab found in green. They're these myeloid cells that are bad guys, right? And this survival curve shows in red, if you have more of them, the cancer is more aggressive. If you have less of them in blue, the cancer is less aggressive. So we think these are really the ones to go after. And we think by going after these, we can make all types of immunotherapies work better, including cell therapies. So this is one of the directions that our lab and many others is definitely headed. So we have a fun group of trainees, both current and former, in our laboratory that just published this really awesome paper in the Journal of Immunology, which describes our efforts to go back to the laboratory and in a mouse model, develop ways that we can mimic the clinical human situation. So we have Megan Whitling at the top, who's an MD PhD student, in my close collaborator, Dr. Crystal Paulos' lab, and then Dr. Francie Bennett and Dr. Emily Warren, who were surgery residents that worked between our labs, specifically on cholangiocarcinoma. Dr. Warren was actually supported, and I'll talk more about that later, through a generous donation by the Cholangiocarcinoma Foundation for a fellowship. And together they worked to develop a system where we can, and I apologize to the animal lovers out there, but these are well-characterized models where we can implant, much like patients who are afflicted with this disease, a tumor in the liver. We can take it out and we can identify the T cells and expand them in culture and then give them back. And we know the composition of these tumors, the different types of white blood cells that are present. And then importantly, in an assay where we put them head to head in a test tube together, the lines, things going up higher means there's more killing. These T cells derived from these animal tumors that mimic patients can indeed kill cholangiocarcinoma tumor cells in the test tube. So we have a really cool model that we can now manipulate and do things to and answer experimental questions with the hope that we can make life better for patients and devise new cellular therapies. So what are we doing with this model? We're testing ways to enhance it. And again, we're going after two main things. The first is going after these hostile myeloid cells that we think limit the anti-tumor function. And the second thing is we're trying to essentially make a better T cell to put in. And we have a number of ways that we're approaching that. So one of the other interesting things our lab has stumbled on, my talented lab manager, Jacqueline Hammonds, and my awesome MD PhD student, Alexis Mack, there's been some data published that when patients undergo this making room or lymphodepletion process ahead of cell therapy, there is a rebound. The cells eventually come back, right? They're not going to stay cell-free forever. The bone marrow continues to pump out new cells and they circulate and go through the body, and you want to achieve homeostasis. But the funny thing is that the first cells to really come back in patients are these bad guy myeloid cells that we're thinking are the ones that are inhibiting the responses. Things that you want to protect you from infection, for example, like B cells, they take a bit longer to come back after you receive these drugs or get irradiation. And they've used these animal models to model the same thing. And these dots essentially represent cells. The red is animal that has undergone this lymphodepletion like a patient would. And the red bar means there's more myeloid cells, more of the bad guy cells coming back. So we can essentially mimic this in an animal model and then study ways to interfere with it to hopefully make cell therapy better. So we have systems, again, where we can get the lymphocytes out of the animal, we can put them back into a tumor-bearing mice, and then we can block the bad myeloid expansion and hopefully make these cell therapies work better. So this work's going on in our lab as we speak. We're very excited about it. and we think we can put a dent in one of the barriers to getting the cells to work better. The next thing we're doing is making a better T cell. And this is a really highly collaborative NIH funded project, actually funded in pancreatic cancer, but the cells can be used across many types of tumors, including cholangiocarcinoma. So my colleague, Dr. Palos, her lab has really discovered this new supercharged subset of white blood cells called T cells that express this protein called CD26. And I really equate these cells to being a Swiss Army knife of white blood cells. They have all these different functions. They make tons of immune factors. They can kill. They can, spit out different immune hormones. And really, when you put them back into a tumor-bearing host, they can obliterate it. It's really quite impressive. So we think these are the ones to start with. And then in collaboration with Dr. Ned Waller, one of our other investigators on this grant, we can brew these cells up outside the body using an inhibitor that keeps them with these youthful, non-exhausted properties, and then we want to put them back into patients. So we have a great group of people working on this. Dr. Alashay, our medical oncologist, who's a GI cancer doc, Dr. Yuan Liu, our biostatistician, and this is a multi-institutional effort with Carl June, who's really a pioneer in CAR T cell therapy at UPenn, and we're working closely with their team to engineer these cells and then get them into patients. So just really quick, not to bog down in the details, but this is a paper a long time ago out of Crystal's lab showing the potency of this novel cell subset that we want to engineer. You can see these immune factors made in all the colors over here. Long story short, they make many of them at the same time, so they're very potent. And then when you put them into an animal with a tumor, the good cells that we're planning to engineer are those shown in red. They essentially shrink the tumor. We're looking over time at tumor growth, and they shrink the tumor much better than other types of cells that we've engineered that we thought would actually be the better ones. So a bit of a surprise, but now we're sitting on what we hope is a gold mine with a really functionally powerful T cell that we can direct towards only cancer and then put into an animal or a patient. And we're gearing up to do the final studies that we'll submit to the Food and Drug Administration for what's called an IND application that allows us to go into patients. And this is a busy slide, but it sort of outlines the schema that we're planning to go forth, in this case, pancreas cancer patients with CAR T cells that are directed against antigens on the pancreatic cancer cell. We're giving the funky ones that we think are really good alone, or we think they'll also provide help to the more conventional CAR T cells. And the goal, once we know that they're safe, is to co-infuse them into patients and hopefully make a dent in another dreaded disease. And this process can obviously be expanded onto cholangiocarcinomas that express the same antigen as about 30 to 40% of them actually do. So we're quite hopeful and working very hard in this effort, and hope to have an update in the next several months as this moves forward. So just to kind of end it as well, we know outside of pancreatic cancer, we can isolate and program T cells or other lymphocytes from other tumors, including cholangiocarcinoma. So in collaboration with the Palos lab, this is a lot of work done by a talented research scientist named Megan Wyatt in the center of us here, where you can get out a tumor, you can isolate the TILs and sort of expand them up the conventional way using an immune hormone called IL-2, or we have a novel method with sort of a cocktail of factors we put in with the lymphocytes that makes them way more antigen specific. And we think these guys are going to be better at fighting cancer. So Crystal's lab, in collaboration with our team, have done this in, to date, 16 melanoma patients. We've compared the new cocktail that we have in the cells, and we've shown it makes them grow more and get better properties that we think will be better cancer fighters. And to date, as of today, we've done this also in 3 separate tumors from cholangiocarcinoma patients under our IRB approved protocol, where we can expand these out in the lab, again, from patient tumors, and show these cells up in this corner, 60% of them are those that selectively, we think, based on the markers on their surface, may recognize antigens on those patient tumors. So we're really working hard on this and trying to develop a potent cell product that could, again, go to the FDA and get approval to move forward. So in the future, we have a number of objectives. We want to build a better white blood cell again, starting with other templates like these CD26 ones I talked about, find different cocktails we can grow them with to increase their reactivity selectively against the tumor. We want to use drugs and other genetic strategies to make the T cells younger and thrive more in these hostile environments. And then finally, in the blue, we want to go after these bad immune cells that we think are stunting the activity of the T cells. And we have a number of molecules and drugs and collaborations with industry partners that we're investigating to go after and see what we can do, hopefully with the goal of getting that into the clinic. So again, thank you to the foundation. This is a picture from last year's annual meeting in Salt Lake that we love to go to every year. And then thanks also to the support from the Mather family a few years back to get Dr. Warren off on her own career and plant the seeds for a larger program of cell therapy research between many of our labs. And again, thank you to the Bloom family for sponsoring today's seminar. Really quite an honor to tell you our story and some of the ongoing work we have. And then, this work couldn't have been done alone. We've had a nearly 7 or 8 year collaboration with Mark Yarshwin and Nilo Azad at Johns Hopkins. Still friends and collaborators with Dr. Shishir Maithel, who's relocated for a really wonderful leadership position at Northwestern, although we miss him. And then Dr. Paulos, who you heard all about, sort of our brother-sister lab here at Emory University, and we're co-training many people going after cholangiocarcinoma and a number of solid tumors, and then a talented young surgeon scientist, Mohammed Zaidi, who's committed to a career in cholangiocarcinoma and working closely with us to adapt ideas into the neoadjuvant setting for this disease. So most importantly, we're very grateful to all the patients and their families for providing tissue and enrolling in clinical trials, as I hopefully have convinced some of you that those energies have led into greater research, and we're actively working forward to try to make progress in this disease and doing it as a team across the country and even the world. So I hope there's some time for questions, and I see some comments in the chat. So thank you for your attention.
Thank you so much. Yes, there are some questions in already. So if anyone else has questions, please enter them in the chat or Q&A. I don't know if you can answer this, but how is the current administration's budget cuts impacting your research?
Yeah, so it is definitely devastating to biomedical research what is occurring at the federal level with what we had traditionally been used to working with. It's challenging to operate in an environment where you can't necessarily grow and take risks. So I think a lot of us are in the mode where we're forced, essentially due to fiscal constraints, to really play it safe and not overstretch our budgets. Otherwise, we would go out of business. So we absolutely can't afford for that to happen, but we're getting creative and trying to do our best to seek other sources of funding, whether it be from industry, philanthropy, and really focus on what we can control. And that's just continuing with the limited resources that we have to do the best science and impact patients and engage the community so that there can be strong advocacy across the nation in all circles, whatever your political lean is, that there is importance in cancer research. And this is definitely something that is not wasteful and is impacting patients and worth a sustained and regular, dependable investment in.
Okay. How do you see that CAR T sits in a set of weapons for cold tumors like intrahepatic cholangiocarcinoma against other cell-based therapies for solid tumors, i.e., is it at the top of the list? Can we layer these up one after each other?
Yeah, I'm gonna stop sharing the screen quick, and then I'll be able to better answer the comments if that's okay, but happy to share and pull any slides if people want. So this is a great question. I think there are definitely differences in cholangiocarcinoma with how hot or cold the tumors are. Um, in much of this is actually influenced by the genetics and the DNA within those tumors and what mutations are there. That actually has a good level of crosstalk with the cancer cells and the resulting immune response. So if you're talking about the TIL strategy, if you get a colder tumor, you're going to get fewer TILs, and maybe a CAR T approach would be better because you're actually engineering cells, recognize them and putting them back into an environment where they need more cells. They're not naturally there. So I think this idea of layering them is really interesting. We have to, of course, since many of these therapies are not approved, get the basic safety data and make sure it's okay to put them inside the body before we design really ambitious trials with multiple cellular options. We're not quite there yet as a field, but as we get some initial safety and tolerability data within them, I could definitely see the field going that direction.
Okay. Will any of these therapies be safe for someone who has had nephritis due to traditional immunotherapy?
With the caveat that I am not a physician that cares for patients, typically when individuals will enroll in a clinical trial, it's always best to consult your medical oncologist and care team for any sorts of risks that, would not be a good idea to go on an experimental drug that may cause a autoimmune type reactivity. This could indeed be one of those cases. I can't comment on the specific, you know, uh, scenario that you may be in, but it would be a situation worth having a really careful and thoughtful conversation with your care team to decide that.
Perfect, thank you. For cholangio patients without one of the genetic targets you mentioned for CAR T cell therapy, such as HER2, how realistic is this therapy in a time frame that would be relevant to someone's care?
Yeah, that's an excellent point, and I didn't cover this, but I'll say it because it's kind of the elephant in the room and everyone knows it. One of the other challenges with cellular therapies in general are two things. The first is cost. It's no secret that they're at the early stages and they're very expensive. The second is time. The FDA approved melanoma product from a company called Iovance, for example, it takes on the order of almost 2 weeks to get out a tumor, send it back to the company, and expand enough cells to send back to put into a patient. So the good news is that there's a concerted research effort at many both industry companies as well as academic institutions, including folks at our own place and our partners at Georgia Institute of Technology in downtown Atlanta that are addressing some of those manufacturing and engineering barriers to make the process cheaper. One way people are doing that are exploring what's called off-the-shelf cell therapies. So in essence, you would have a pre-made cell therapy from really any patient that has been engineered in such a way that your body wouldn't react to it as foreign, and you could essentially get the therapy much cheaper and much faster. The other way is playing around with different expansion protocols. And one of our collaborators in the Palos lab, they've actually gotten some of the preclinical work down to a 3-day expansion as opposed to taking cells out for 10 to 14 days. So you cut off a week and a half there, that cuts cost, it returns it back to the patient faster. We're not there yet, but a lot of the earlier clinical trials that are just getting going are, and even some of the companies that make cells are exploring shorter expansion times. And in fact, the cells may be better too. They may be less exhausted. They haven't proliferated as much. They're not as tired. They're a bit younger when they go in the body. So I think the jury's out on that, but that's something that we're definitely looking into.
Okay. Can T cell therapy be also effective against tumors with microsatellite status stable and TMB low?
So, in theory, yes. That, as you know, for those that may not live in the space, these particular mutations make a tumor typically more immunogenic. They'll express more antigens. They'll typically have more immune cells surrounding their tumor and being able to activate them. And as a result of that, when you give these immune checkpoint antibodies, patients with these microsatellite unstable tumors will respond very well. It's the converse for the tumor type that was posed in the question, the microsatellite stable. They're typically more immunologically cold or inert, meaning there's fewer immune cells living in that tumor. And as a result of that, when you give these immune checkpoint blocking drugs, there's really no targets to hit. There's nobody home. There's nobody for the antibody to go in and bind to. So you're less likely to have an impact. So that, theoretically is one of the ways in which engineering a cell from the circulating blood outside the body, infusing it in the vein, and putting it back into a patient could be quite impactful because you're circumventing that natural environment of the tumor to lack immune cells, and you're sort of like hitting it with a sledgehammer and willing them to go back in. So whether or not they survive and persist is another story. That's a barrier I tried to touch on, but I think that's potentially one of the advantages of cellular therapy is that patients with these types of tumors could indeed engage immune responses if you have a good antigen on the surface to really go after. And let me know if that answer wasn't clear. I hope I did it's kind of complicated with all the things going on.
Are there any signals demonstrating that specific intrahepatic cholangiocarcinoma mutations respond better to TIL or CAR-T?
Too early to know. I'm not aware of, a really robust, rigorous trial that has had enough patient numbers and really looked in depth at what might be present on those tumors from a mutation standpoint or not, and how they ultimately do in response to the cells. But fortunately, the era of genetic testing is upon us, and hopefully, most patients that go in, if not all, get their tumor genomic information, and that data is on file with the physician. So, in retrospect, hopefully, we can go back in time and look at that data as an aggregate for multiple trials as we do more of them and maybe make some connections and then test them definitively in the future about whether there's a relationship between a gene alteration and then response to one of these cell therapies.
Okay. In your experience, do you know if age is a factor? So do younger patients respond better?
So with cell therapy, we don't know. I would say it's the immune system. The younger you are, most times it's a little less affected by age. There's some literature out there that might indicate, telomeres, which is a specific portion of chromosomes and DNA, they shorten as you age, and it results in various downstream biologic changes that may be immune in nature. But with that said, I mean, in some of the heme malignancies, the leukemias, for example, CLL, this is a disease of older individuals, and many of these patients get CAR T cells and they have complete responses and do quite well. I know the blood cancers are absolutely not cholangio, but the immune checkpoint blockade antibodies are another example of an immunotherapy that can routinely be given to older patients. And one of the most famous examples, fortunately, is out of our institution. Former President Jimmy Carter was treated in his 90s by one of my colleagues who's a medical oncologist with these PD-1 blocking antibodies, and as you know, did very well with his melanoma. So I don't think age is the only factor, and it shouldn't deter someone from investigating this as a therapeutic option should it be deemed safe by their treating physician.
Okay. Do all cells expand for patients? Do you need the same amount of cells to reinfuse to be successful?
Yeah, so no, they don't. We do pretty well in our hands, especially with the cholangiomas and with the melanomas. Most tumors we've had, we've been able to grow out cells, and other groups have this sort of experience. And there are tumors that are just so devoid. Some of the pancreatic cancers, for example, that we've worked with in our lab, they have very few immune cells, and it's hard to get anything out of them. So it's not a guarantee, and there is a little bit of finesse that has to go into it. And we're working our best to identify the optimal way to get as many cells as possible. Usually, when you write these clinical protocols to answer the second question, you pre-specify ahead of time the number of cells you need that you're going to give back to the patient. So that's usually worked out ahead of time. It's in the millions of cells. It's quite a lot of cells, but in the majority of cases, these tumors can be manipulated in a way that you can get the TILs out of them and expand them and have a product for most patients.
Okay. Who is cellular therapy not good for, such as someone with autoimmune history?
Um, yeah, I think in a majority of the clinical trial eligibility criteria, those are often exclusion criteria for a variety of immune agents. Although there are some studies, even with the earlier immune checkpoint blockades, looking at even patients with like transplants, if they're on immunosuppressives, can they get the drug and still have a transplant, or still have the transplant take? For the cell therapies, some of the trials, patients with brain metastases may be excluded. Obviously, performance status is good. The lymphodepletion regimen can be pretty rough, I'll be honest. Some patients don't tolerate it as well as others, but in many cases, there's at large centers, including ours, there's care teams that they have their own working group that manage patients that are on cell therapy trials only. So they really know how to uh, allow the patient to get the supportive care they need to continue on and go through with this to potentially gain the benefit from it and make it as tolerable as possible and fewer toxicities.
Okay. And then one last question, because I feel like people are asking about clinical trials. Do
Sure.
you run trials or are you just doing the basic science piece?
So yeah, my training is in the laboratory. I'm a PhD, so I do not see patients or treat them on trials. With that said, our group and many others work as a large collaborative unit where we approach problems like cholangiocarcinoma as a multidisciplinary team. So we have daily communication with the oncologists who care for the patients on the trials, and there's a bi-directional communication between myself, my team in the lab, the clinical team that's taking care of the patients, as well as other collaborators in labs that, all work in tandem to ensure these things go well. So we are integrated, and I really see our role as giving the physicians ideas and giving them tools in their arsenal to hopefully help you as the patients have new options and have hope as we move forward with these really horribly aggressive cancers.
Perfect. Thank you. I know we're at time. I want to respect your time. So thank you so much for your presentation, answering our questions, and thank you to everyone who was on the call, who listened in and sent in their questions to us.
Thank you so much. It's really been a pleasure.
Thank you. Hope you all have a good day.